I didn't expecting them will be talking talking at the same place, so they basically work on the same kind of project. Instead of ovary cancer, Tony is working on the breast cancer. It's just much bigger library, which contains like 4,000 of SNPs associated with breast cancer. That's it. But about Tony, so he didn't break it down. I didn't take apart the reading. But I find that he actually is very persistent. Okay, this is so another big quality for being a scientist. Okay, you can tell by the last stage he's still working on his bench and doing the lab work. And then I think this is from his training. He is a member in the field and track team and doing long range running, right? So for long range running, I was in the track team, field track team. I'm high jumping. I hate long range running because it's so you can get so tired. So I don't have that much persistence, but he has, and that's why in the lab he can work every day, very seriously working every day from the beginning to the end, and from the first day to the last day. Okay, so that's the good part. So thank you.
Thank you, Dr. Lee. So before I begin, I'd just like to thank the Hillman Cancer Center for giving me the opportunity to present my data and the rest of my presentation, introduction, everything. And so with that being said, my project is called Identifying Functional SNPs Associated with Breast Cancer Based on GWAS Data. And so before I get into the introduction, I'd like to ask, how many of you out there know someone that has had or has cancer, or if you yourself have or have cancer? So a lot of you. So cancer claims the lives of 600,000 people a year, and another 1.7 million are diagnosed. So it's a very serious problem. So with that being said, cancer is an example of a complex disease. The reason we don't have a cure is because complex diseases are difficult to study and treat because they're associated with effects with multiple genes, as well as environmental factors. So environmental factors such as obesity, multiple genes. So there's not like one gene we can pinpoint and say this is the one that causes the disease. There's multiple genes, and for that reason it's very difficult to treat and study. And so, however, with the introduction of genome-wide association studies, or I'll be referring to it as GWAS, it opens a new window for studying these complex diseases by identifying numerous disease-associated single nucleotide polymorphisms, or SNPs. And so, here's a diagram of GWAS. And so we have 5 different case SNPs, 5 different control SNPs. And so how this was conducted is that we have thousands of different subjects that have the disease and that have the control. So they were matched in order to eliminate any confounding variables. And so age was matched, gender was matched, and et cetera. And so SNP1, and so how do we indicate it? It's with the case over the control. So for example, SNP1, there's 5 instances here compared to only 2, so we can assume that this is associated with the disease. Same with SNP2. Now SNP3 is the same, so it's not necessarily associated. Same with SNP4 and SNP5. And so the associated SNP is considered to mark a region of the human genome which influences the risk of disease. And so, as you can see here. And so here's another diagram. So this is a region of the human genome. And so we have gene 1 and gene 2. And so we have a lead SNP for GWAS analysis that's tagged through GWAS. And so this is how GWAS works. So it tags a lead SNP. And so that lead SNP represents multiple other SNPs in the same linkage disequilibrium. And so you can see the red diamonds here. And also these green ones are the SNPs not in the linkage disequilibrium, so they're not represented by the lead SNP target for GWAS analysis. And so SNPs can have, as the name implies, they can have multiple different bases. So it's an A or a G here, a C or a T. Now this one, we don't know what it is. It could be a C or a T. So that's why it's not in linkage disequilibrium with the lead SNP. So we have to identify which SNP is actually the functional SNP. And so this is the lead SNP. So if we identify as functional, if we identify as associated with the disease, we don't know necessarily if it's the functional SNP. And so we have to identify the functional SNP that's actually associated with disease. And the other ones may not be, so we just have to conduct experiments to do that. So here's our experimental approach. And so we first cultured our breast cancer cells in the RPMI 1640 medium. And so what we'd have to do is daily we'd have to split those cells in order to help them grow more. And then we would transfer them to multiple petri dishes. And then once we had enough cells, we needed like 10 or so plates of each different cell line. And so then we would once we had enough, then we would isolate the nuclear extract from the 5 breast cancer cell lines. And so we had 5 different proteins. Their scientific names are like MDA, MD 468, and it was MDA, MD 231, MDA, MD 453, and then there was BT 474 and MCS7. So those were our 5 breast cancer cell lines. Say was doing ovarian cancer, and so he only had one. He only had the OV8 protein. And so the difference between breast cancer and ovarian cancer, my project, CS project, was that there were 5 different cell lines in mine compared to only one in his. And so also we would obtain the breast cancer RealSeq library that would be from the company. And then what we would do is we would use PCR to amplify that RealSeq library. And then with this nuclear extract that we isolated from the 5 breast cancer cell lines and the amplified DNA that we received from the company, the RealSeq library, then we would use it to perform electrophoretic mobility gel shift assays, otherwise known as EMSA, with the DNA from the library and the nuclear extract from breast cancer cell lines. And then that would be how we kind of separate the functional SNPs from the non-functional SNPs, and I'll get to that in a minute. But then we would do 10 cycles of this, and so we would do so we first do this, and then we go back to PCR, and we do gel shift assays again, and go back to PCR for 10 total cycles, and then at the end of the 10th cycle, we would get our results. But here is the principle of RealSeq, and so we have our we have the 31 base pair SNP sequence. So this would be the our PCR here. So we have the 3' primer and the 5' primer. The we have 2 different alleles, the risk allele and the non-risk allele. And so we would amplify this DNA through PCR so we could analyze it then through EMSA, or electromutation, EMSA for short. So here's step 1, here's step 2. We would do EMSA, we would do 10 cycles of this, like I mentioned. So how we would do this, we would have a controlling without nuclear extract, and so this would just be the DNA. Say you mentioned something similar. So this would just be the DNA here, and then we would put the nuclear extract here with the DNA, and then we would see this gel shifting. We would see the bands located in different spots, and so here we have it down here, and so this would be what we analyze. And so we have the control, we have all this DNA. In the sample, we see a little bit less in each of them. And so we have to analyze the ratios of the control SNP1 to the sample SNP1, and then the control SNP2 to the sample SNP2. And so if the ratios were the same, then we can say that that SNP is not functional. And so it would not necessarily contribute to the disease. But if the ratio is different, however, so in SNP2, the ratio was 1 to 0.25 in the sample to control. And so therefore, since the ratios are different, we can therefore say that that SNP is functional. And here we have, down here we have risk, non-risk, risk, non-risk, risk, non-risk, this way. These are just the regulatory proteins in nuclear extract. It says that down here, but this thing is blocking it. So then, this is the outline of the RealSeq screening. So the difference between my project and Say's project was that with breast cancers, there's a lot more SNPs, as Dr. Lee also mentioned. Say's project only had like 800 so SNPs. There's 4,316 SNPs with 8,676 SNP sequence, both risk and non-risk alleles in the breast cancer. And so what we would do to narrow that down, we'd have 10 cycles of RealSeq, next generation sequencing quality control. We would narrow that down to 3,436 SNPs. And then eventually, from analysis the results, we get 1,720 candidate functional SNPs with allele-specific gel shifting in cycle 10. Now, I do not have all 1,720 candidate SNPs. That would take forever. And so what I did in my project through 6 to 8 weeks is that we have 5 SNPs to show you to illustrate the concept. Now, first, I would like to show you the results of the PCR. So this is an example of one of the PCR results we did. And so here we have the control 1, sample 2, sample 3, sample 4, sample 5. And so that we had 3 bands for each sample plus the control. And as you can see, all the bands here are visible. So therefore, we know we had the right amount of DNA for the PCR. So this is the gel. We ran through an acrylamide gel to visualize the results of PCR. And so we have the bands at about 100 base pair, I believe. And so since they were all visible, we could move on to gel shifting. And so as I was mentioning here, we have 1720 candidate functional SNPs. And here is the 5 SNPs we were able to do to illustrate the concept. And so as you can see here, the bands are only, you can see the bands there's like a difference in visibility across these different alleles. So the C allele, you can mark by these red arrows. You can see it's fainter here, but it's stronger here. Same here, stronger here, fainter here. So that difference in bands indicates that we are working with a functional SNP. And so because of the difference of bands, that indicates that the amount of protein that binds to that allele at a certain point is different. And so this is the gel. So the larger the size, I should say the smaller size, the further it will go down in the gel. And so with the protein, so if it's more of the protein for one allele is located higher up, we can therefore say that a greater mass of protein binded to that allele. We don't know if it's a different protein or just more of one kind of protein. We just know that greater mass of protein binds to that allele. If the band is more higher up than the other allele. So you can see here, we're marked by all those arrows, a little bit stronger here, stronger down here, and you can see the comparisons. So those are the 5 SNPs that we analyzed. And so our future directions would be to validate all 1,720 functional SNPs. And so like I said, we were only able to do 5. And so we have to validate all 1,720 by gel shifting performed individually. And then we want to also screen the same breast cancer library with the nuclear extract isolated from the 5 different cells, representing each of the 5 different subtypes of breast cancer that I explained earlier. And then we also want to identify the protein that's binding to each of the functional SNPs, so we can perhaps develop a treatment for breast cancer, because that's the ultimate goal. And so I'd like to give a few acknowledgments. So I'd like to thank Yihan Zhao, our mentor, Seyun, and my mentor, Dr. Gong Li, for letting me work in his lab. I'd like to thank CU for letting me come to all the lectures and for really getting me involved with this program. Dr. Andrew Duncan and Dr. Pete Alexander for making my project and presentation possible and for being awesome site heads this year. So that's about it. Questions? Sophia?
Could you go back to the previous slide?
Yes.
Your last goal, do you know how you would go about doing that? Like identifying the protein binding to each of these functional SNPs?
That's a good question. There may be some sort of, I don't know the exact technique that we would use to undergo it, but it would probably have something to do with like marking the protein and kind of identifying the characteristics of it. So like how much of which component is involved, like which amino acids are involved in the chain, and we would have to analyze the structure and everything, but I'm not exactly sure what technique we would use.
You want my help?
Sure, if you know how to answer that.
That's a great question, because we can see there's 17 countries different, right? We cannot do one by one piece, right? So we developed a very high efficient technology. We can isolate these protein binds with specific SNPs within like 1 or 2 days for each of these based on the protein binds to it, and then we use the control that's a different sequence, so without the SNPs, protein will not bind to it, and then we can do it parallelly and say anything binds to this SNP, not this one, this SNP binds. It's complicated, but it's explaining the stuff.
Any other questions?
Thanks.